Okay, so welcome to the second video in our DNA RNA module. In this video, we're going to specifically look at DNA replication, and we're going to look at how DNA is able to duplicate and replicate itself, which is very important whenever cells divide and reproduce. And this process specifically exists in um, cellular division processes such as mitosis and meiosis. And it's really important for that DNA to duplicate because when a cell reproduces, one copy of the DNA will go to the new offspring cell, while the other copy will be maintained kind of in that parent cell. And if you remember, there were pairing rules that were discovered when Watson and Crick figured out the structure of DNA. And so each strand of DNA basically serves as a guide for the reproduction of the other strand because there are specific rules for the pairing of these nitrogenous bases. And if you so what that means is if you know one base, you automatically know the other base that it's paired with, because adenine is always paired with thymine and vice versa, and guanine is always paired with cytosine and vice versa. So if you see a G, you automatically know that the opposite base pair is got to be a C. And if you see a T, you know that that has to be paired with an A. So if you have one strand, you automatically know what the second strand is. And so this makes DNA replication really sound because of these base pair rules. So for example, if you have a parent DNA molecule, that molecule will basically be separated using, the body does this using enzymes, what's called DNA helicases, I believe, which is what unwinds that DNA molecule. And it basically goes in and separates those hydrogen bonds. And when it separates those hydrogen bonds, you now can use each parent strand to serve as a template to build a new daughter strand. And this process is called semi-conservative replication, because whenever you get your finished product, the daughter DNA, one half of that molecule is the old parent strand, and the other half of the molecule is the new daughter strand. And again, so this is what's known as semi-conservative replication. And what actually builds these daughter molecules are several different enzymes. So for example, your DNA polymerases are the enzymes that will go in and make covalent bonds between the new nucleotides. So the covalent bonds between the nucleotide of the daughter strand and the old parent strand. So those polymerases bond those nucleotides together. They also will go in and help repair the DNA that could be damaged. So let's say it's damaged and maybe it doesn't have the correct base pair. Maybe something happened and there was a mistake. Those polymerases can go in and fix that. And so what happens whenever a DNA molecule is replicated, you basically have these spots that exist at several places throughout the DNA molecule. And these spots are called origins of replication, and these specific sites. These origins of replication are basically where the DNA becomes unwound initially. And it creates what's called replication bubbles. Okay, and you'll see that exactly what we mean by that in the next figure. And so what this DNA replication process basically does is it ensures that all of the cells, all of the somatic cells in an organism are genetically identical. And so that genetic information is passed from parent cells to daughter cells. And so you also get this information being passed to the appropriate offspring. Here is a figure looking at this replication process. In this figure, we have our original parent strands, or original double helix. We see that there are two origins of replication sites on this parent strand. These two origin of replication sites is where the DNA molecule is going to basically start unwinding or separate initially. You'll have an enzyme that will go in and will separate those hydrogen bonds. And what you end up with is this replication bubble. Now you have this area that's separated, and your DNA polymerases will come in and they will start replicating and adding new nitrogenous bases that are associated with the corresponding base. So for example, if this first base on the parent strand is an A, then the DNA polymerase will add a T. If it's a G, it will add a C. If it's a C, it will add a G, so on and so forth. And the replication bubbles will replicate in opposite directions. Okay? So basically you have replication going to the right and to the left, and if this bubble is doing the same thing, then eventually they'll meet in the middle. And so this speeds up that process of replication. And then afterwards you end up with two daughter DNA molecules, where each daughter molecule has a old parent strand and a new daughter strand. So again, semi-conservative replication. Okay, so that's it for the replication video. In the next video, we are going to look at how DNA is transcribed into RNA. And then we'll look at how RNA is translated into protein. So we're going to go from this information source, which is DNA, to a specific working protein that may determine an organism's hair color or eye color or feather color or height or its muscle tone, etc. Right? And so ultimately the expression of these proteins, which comes from the DNA information, has to go through this process of transcription and translation. And so the next video will look specifically at transcription, uh, so we can understand how we get DNA to be transcribed into RNA molecules. We'll see you in the next video.